So your last test is the 6th, which is the last day of class, right? Which is Thursday. My last office hours for the semester, I don't have office hours during finals week. It's way too late to talk about anything during finals week. Let's be serious. Okay. My last office hours are the 7th, generally all day, so that you can look over all of your tests, including the one that you took on the 6th. You might not have the short answer, but you will definitely have the bubble sheet part and a key for that. Okay. So all of that will be, and then ready on the 7th, and it's usually here, and it's usually like 10 to 2 or 10 to 3 or something like that, okay? Your final is the 13th, 9:10 to 11:10. Please note, I can't change that time or that day, so if you usually like work after this class, you need to make that block available. The final is 100 questions. It is comprehensive, okay? Now, let's revisit things that we haven't talked about since the beginning of the semester. Under what circumstances can you skip the final? Yeah, and what is an A?
92.5.
92.5 or above. Okay. 92.5. Do you email me if you have a 92.4? No, Karen, you don't, because somebody in here is working 70 hours a week and has 4 children, and they're not emailing me, so shut your pie hole, okay? Do not email me. It doesn't matter who you are. Even if you were Mike, who I love, he's not going to need it. But if he emailed me, I still would delete his email. My husband actually deletes those for me because I get all stressed out. And I'm like, this one's really sad. And he's like, let me do it for you. Delete, because he doesn't care about you people. Okay. All right. So that's your final. And just to remind you, if you do an evaluation for your class, your grades show up faster. That's how they're rooking you into doing it. Did you know that? What? I think they just did. So if you do the course evaluation, as soon as I post your grades, you get to see them. If you don't do a course evaluation, you have to wait until all grades are posted to see them. Go ahead.
Final in this room.
Yeah. It's in here. I feel like there's something I'm forgetting to tell you. What is it? Oh, liver paper, 5% bonus on your final. If you come in and bring your liver paper. Oh, that's it. I wanted to tell you the whole final philosophy. Okay, here's the deal with the final. We talked about this at the beginning of the semester. I'm going to reinforce what happens with the final. Okay, so there's only one circumstance in which you can stay home for the final. Okay, and that is if you're already making an A, because I think you've earned it. Personally, it's not easy to make an A in physiology. Guess what? Guess who made a B+ in physiology as an undergraduate? Me. Okay? I was also a total slacker, but whatever. Unrelated. If you make an A, you don't have to come. If you don't make an A and you don't come, I will enter that 0 as a grade. I won't drop it. Okay? Because here's the philosophy. If you haven't gotten your A, you need to keep working to try to get your A. If you come and take that final and you make a better score than one of your midterms, I'll drop that lowest one, right? Okay. What if you do worse on your final? I'll drop the final for you. Okay. So it cannot hurt you. If you have taken all your tests, you can go into your final and you can't go down, but you can go up, right? So you got to keep working until the end, unless you've already gotten your A, and then you can sit home and feel superior.
I don't like it. Are the lab grades going to be added in before, so we know exactly?
You might know your lab grade. You'll probably know your lab grade before I do because they have to come through channels to get to me. So you can ask Joe, hey, what did I make? And then, mentally figure it out in there. The lab grades have to go from the lab instructor to the lab coordinator through me and then to me. So it takes longer to get to me than it does to get to you. Good. You should be able to see it on your Canvas, right? For your lab? Yeah? Okay. All right. So any other questions? Everybody good? Okay. So I think that's it. That's all right. Okay. So we are going to do blood today. It's not outrageously hard. It is one of those things that seems really complicated at the beginning, but then it clarifies, and it's nothing like cardiovascular systems, so don't worry about that kind of complexity. But if you get confused, just wait a minute and I'll simplify it for you. Okay, so before we get into blood specifically, let me just talk to you guys about how I approach blood in physiology. A lot of times you get blood multiple times. You get it in anatomy, and then you get it in micro, and then you get it in phys. So I concentrate on the stuff that you don't get in the other two classes, which is blood clotting. So you usually get immunology in micro, right? And you usually get the basics of blood and anatomy. So I'm going to do the thing that's important for clinical situations, and you don't get other places. So the other stuff I'm just going to go through relatively quickly. Of course, you guys remember that basically you've got erythrocytes and leukocytes and thrombocytes, right? With your erythrocytes, we're going to talk about them a little bit in this set of notes, but then we're also going to talk about them in respiratory system because, of course, their function with carrying gases, right? The primary function of an erythrocyte is to carry oxygen around and also a little bit of carbon dioxide around the body. So we'll come to those in just a second. Leukocytes are, of course, white blood cells. Just briefly talk about those before we move on. They're bigger, they're less numerous than red blood cells, and their primary function is twofold. Defense, which is what happens immediately upon invasion of some kind of pathogen, toxin, and immunity, which is preparation for the next time, right? Defense, immunity. And each of the leukocytes has like a different specialized function. Like some of them respond to bacterial infections. Some of them respond to viral infections. Some of them are allergies, like basophils. Basophils make histamine, which is involved in the allergic response. So do you guys know the mnemonic for which ones are most abundant in order? You don't know never let monkeys eat bananas? Yeah, never let monkeys eat bananas. That's the normal proportions. Never neutrophils, let lymphocytes, monkeys, monocytes, eosinophils, basophils. Those are the relative proportions. Okay, and then we're going to spend some time today talking about thrombocytes, which are platelets. So platelets are not actually whole cells. They are actually cell fragments. They were made from red bone marrow, but basically by the time they're done, they're just kind of a bag, a cell membrane bag that's filled primarily with clotting proteins. Okay, and we're going to talk about their function. And then last but not least is just talk about blood plasma. Okay, so blood plasma is, of course, the extracellular fluid that is around your blood cells and your platelets, and everybody knows that. And it's about 90% water, so anything water soluble is really easy to transport in blood plasma. What kinds of things are water soluble? Well, we've spent a lot of the semester talking about like transporting hormones. Most of those are water soluble, right? Okay. We also know that electrolytes are kind of a big deal. Ions, all of those are water soluble. And wastes and nutrients, any water soluble waste and water soluble nutrient is going to go from point A to point B in the blood plasma. So that's okay. And then the other thing that is in blood plasma are some unique proteins that are in blood plasma. Usually those are insoluble, meaning they stay trapped in the plasma and don't go into the interstitial fluid. The ones we're going to talk about in detail today are going to be the clotting proteins, but let's just briefly talk about the other ones and then we'll move on to Roman numeral 2. Albumin is a plasma protein that is made by the liver, okay? The liver is all up in the cardiovascular system's business, especially anything related to blood. And albumin, remember how I told you that lipid-soluble stuff quite often needs an escort protein to help it get carried around? This is a really common escort protein. And it's in relatively high concentration in the blood, and it's insoluble. So in addition to escorting lipids, it also exerts an osmotic pressure to help maintain the, does that make sense? If it's high in the blood plasma and low in the interstitial fluid and insoluble, then it like helps you to maintain your blood volume. Okay. It also acts as a buffer to prevent pH changes in the blood. So that's albumin. And then globulins, right? Have you guys heard of globulins before? They were literally named globulins because IN means protein and it looked like a glob. They're like, let's call that one a globulin. And you think that people are smart. So globulins. So you've got alpha and beta globulin, which we know the function of, and then you've got gamma globulin. Alpha and beta globulin are also produced by the liver, and they are also escort proteins for lipid-soluble stuff. They just escort different things. Okay, that's alpha and beta globulin. Gamma globulin. Have you guys heard of gamma globulin shots? Usually here in Utah, people have, because gamma globulin shots. Gamma globulin is antibodies produced by white blood cells in response to some kind of pathogen. And the reason people here in Utah quite often have heard of it is that we have a lot of people who serve missions to foreign areas here. So let's say I was serving a mission, which is hilarious because they don't let atheists do missions, but I do have a good, important message for you, though. So what would happen is, let's say I was going on a mission to Uganda, or as in the military, both are equally ridiculous with me. But I'm going to Uganda. I've never been exposed to pathogens that are unique to that particular area of the world. I've been raised in the northern hemisphere, right? North America, haven't really met any equatorial pathogens, and certainly not from an entirely different continent. So what I can do is you can artificially bump up the antibodies that are that region specific, so that when I get there, inevitably when I get exposed to those pathogens, right, I don't get taken down immediately. What it's doing is providing a little immune system buffer for your inevitable exposure. So they shoot you up with antibodies that are specific to an area, and then you go there, and then you spend the first few weeks building up your own antibodies, and by that time the ones that they shot you up with are gone. Okay, that's a gamma globulin shot, and they're specific to specific regions, right? Apparently they're really, really thick and viscous. They call them peanut butter shots, and they hurt like hell when they go in. Nobody's had one here? I almost always have somebody who's had one. But they get less and less necessary as the world becomes more global, and we don't really have super isolated pockets of antigens and antibodies. So that's gamma globulin. Gamma globulins are produced by lymphocytes. And then the clotting proteins that we're going to go through today, most of them are made by the liver. And we're going to talk about this today. Okay, so let's briefly just talk about what blood is like. Blood is literally thicker than water. So I've never particularly liked that expression because I don't know why friends are water. I get why family is blood, but why are friends water? I don't get it. Okay, so it is about 5 times as viscous as water. To me, blood is like about the texture of whole milk. I mean, you've bled before. You know what it's like, right? It's sticky because of all the sugars and proteins that are in it, right? It's a little hotter than your average body temperature, just because it's going through like metabolically active tissues and that kind of stuff. It is really, really tightly constrained in pH. Normal blood pH, in which your proteins are maintaining their structure, is 7.35 to 7.45. Does anybody know the 2 organs that are primarily responsible for maintaining the blood's pH? There are 2 organs that are constantly working on this. What? Respiratory system and kidneys. Yeah, deciding what to pee out, and then respiratory, blowing off carbon dioxide. So we're going to do that as we go through urinary and respiratory at the end of the semester. Let me tell you one thing. Urinary and respiratory, you would like to have more time to absorb the material, but you don't have it, right? We will finish the material, but you will do better and feel less overwhelmed if you preview the material. So when you get tired of your family this weekend, right? That uncle pisses you off again, right? Or you want to go be alone with your mashed potatoes, watch a video, okay? And then you'll be less stressed out when we get through the material. Okay, so 7.35 to 7.45. It's isotonic to about 0.9% saline. It makes up about 8% of your total body weight. Males, just based on body size, usually have more than females. 5 to 6 liters in a male, average 4 to 5 liters in a female. Just primarily body size, really. Okay, but there's also some differences in male blood in addition to the amount. Okay, so what does blood do? You know what it does, right? It takes stuff from one place to the other place, oxygen from here to there, carbon dioxide from here to there, takes hormones from where they're made to their target cells. Also helps regulate your body pH because it's got buffers in it. Regulates ion composition because some places you have a whole bunch of like calcium, like in the bone, and other places you need it, like in the brain, and it helps to do that. Helps regulate your fluid volume of your circulatory system because it's got so much sodium in it and albumin. It is the only fluid that you have that can clot, so it protects you from fluid loss of all sorts. It also protects you from pathogens and invasions of microbes, and also helps stabilize your body temperature. 2 ways. First, it's mostly water, so hydrogen bonding, forming, and breaking helps to dissipate heat. And then also it works like a swamp cooler, right? From wherever it's hot to wherever it's cool, wherever it's hot to wherever it's cool, right? So it helps stabilize your body temperature in that way. All right. So now what we're going to do first, before we get into platelets, is we're just going to look briefly at red blood cells and how you maintain your red blood cells and sort of what they do for you, big picture. And then we reinforce this with the respiratory system, because you have to talk about red blood cells with the respiratory system, of course. Okay, so you guys know where red blood cells are made, don't you? Yeah, they're made in the red bone marrow, right? And then they get dumped into the bloodstream. The primary function of a red blood cell is a gas ferry, not flat, but toot ferry, like carrying gases around in the body. Okay, primarily oxygen, but it also does carry carbon dioxide, which you may or may not have known before now. Okay, so in order to make red blood cells in the bone marrow, you need some nutrients and some hormones in order to do that. Good? Okay, so one of the nutrients that's really important for making red blood cells is iron, because iron is a component of what? Oh my God, hemoglobin. Hemoglobin. It's a component of hemoglobin. That makes me so happy. My husband's like, you are a nerd. Do they actually laugh at your jokes? I'm like, screw you, I'm hilarious. Okay, all right, so here's hemoglobin. I'm sure you've seen hemoglobin before. Okay, the important parts of this are there's a protein portion, right? That's called the globin portion because it's a globby protein. 2 alpha subunits, 2 beta subunits. And then at the center of each one, there's a heme group. At the center of the heme group is an iron atom, okay? Why the hell do you care? This is where oxygen sits in the ferry, okay? Oxygen gas rides on the heme portion of hemoglobin. So look at this molecule of hemoglobin and tell me how many molecules of oxygen gas it could carry. 4, yes, exactly, right? Okay. If you do not have adequate iron, you won't make adequate hemoglobin, which compromises your oxygen carrying capacity, which compromises cellular respiration. Yes? So this is getting oxygen to where it needs to go. You ready? Okay, so in order to make erythrocytes, and that process, by the way, is called erythropoiesis, in the red bone marrow, you have to have iron. Yes? And the problem is that you are losing iron relatively regularly. You lose it in your sweat, you lose it in your urine, and if you are a female who's menstruating, you lose it in your menstrual tissue, right? Because when you menstruate, you lose vascular tissue. I know we think of it as a fluid, but what it is a really vascular tissue that you lose. So it's lost in these ways, but your gastrointestinal rate, your GI tract can change your rate of absorption. And then if you take in excess iron, you store it, and you store it in your liver. Okay, so let's look at this figure for a second. Ready? Okay, so don't worry about T iron, it's just a specific form. So here's what happens, guys. If you have excess iron, you store it in your liver, right? If you are depleted in iron, you dump some of it back into your bloodstream, right? You can also change your rate of iron absorption in your digestive tract. Whenever you actually take the iron, you deliver it back to your bone marrow, right? Whether it was dietary iron or iron that you released from your liver. So you basically have this little storage and retrieval mechanism that's primarily based on your liver. Are you seeing that your liver is really important for storing a hell of a lot of stuff, right? It stores your glucose, it stores your iron, it also stores a whole bunch of your red blood cells. It's just like a big storage tank. Okay, so let's talk briefly about iron. Where do you get iron in your diet? Anybody know? Yeah, meat. Animal products, where else? Because vegans can get what?
Chocolate. It's in dark chocolate.
Is it really?
There's so much, yeah.
Oh, I did not know that. That's exactly why I eat it. What? Leafy green vegetables, cereal, nut, egg yolks, beans, shellfish, meat, liver liver of other organisms is a great way to get iron, also a lot of cholesterol. But so if you are vegan, right, this is one of the things that you need to be intellectually aware of, right? Being a healthy vegan is an intellectual process. You actually need to pay attention to what you're eating. I always consider like being a healthy vegan kind of like being a type 1 diabetic. Like you need to like be concerned about your nutrients all the time, especially if you're trying to raise a healthy vegan child, which is totally possible, but only if you're paying attention to what you're doing. So you're going to need to pay attention to what you eat to make sure that you get adequate levels of iron, or else you're going to be one of those pasty ass vegans with no oxygen carrying capacity. Now liver is kind of an interesting one. In this culture, we don't eat a whole hell of a lot of liver, but in other cultures they do, including in my redneck Southern culture, we ate a hell of a lot of chicken livers. And I was anemic as a child. And my mom used to fry up chicken livers and put them in toothpicks on the bottom level of the fridge, and I'd just toddle in there and get myself livers, which seems absolutely horrible now. I don't think I'd eat liver now, but I'm no longer iron deficient. Okay, you also need folic acid and vitamin B12. These two are primarily necessary for DNA replication prior to cell division, and of course erythropoiesis is cell division, right? So these are a big deal. Folic acid is primarily from green plants, yeast, liver, and citrus, but vitamin B12, it's really primarily found in animal products. But again, if you're a vegan, you can get it from nutritional yeast, but you have to pay attention to what you're eating, right? Go ahead.
You can get vitamin B12 shots.
Yeah, you can get shots as well. Yeah, but you don't want to have to shoot up your children, so you got to figure out what the hell to feed them, right? Go ahead.
Can you build up like an iron toxicity if you eat high levels of, like, say, red meat consistently?
Not usually in women, yes in men, because we menstruate regularly, right? Men can get erythrocytosis, and also, which is too many red blood cells, and also they can build up too much iron. But guess what they use? Guess what they do with men who have that? They bleed them. You just give blood on a regular basis. Just like all you guys need to do is menstruate, and then you'd be totally cool. And then we would feel better about the whole situation. I mean, if you really want an egalitarian society, let's consider it. That's all I'm saying. Okay. All right. So that's vitamin B12. It's found in large quantities in animal products, not just meat, though. Also dairy and eggs and those kinds of things. But also is in nutritional yeast and a couple of other, I mean, like it's primarily only high in nutritional yeast, so you kind of have to supplement with nutritional yeast to raise a vegan kid, right?
Yeah.
Yeah. You probably know tons of vegans, huh?
Oh, yeah.
Yeah. I know a few. Yeah, I should ask Jack how he get. Huh?
Oh, yeah. So
It's in what? No, if you're, yeah, you can't eat eggs though, if you're vegan.
I
Yeah.
keep forgetting that.
I know. I've got a vegan friend who raised her kids vegan, and they didn't even eat honey because the bees were enslaved.
Oh.
I was like, what? Like they're just doing a bee thing, right? Just like take it. Wow. I didn't even know that was a possibility. But I mean, yeah, it's fine. I was just trying to, and then eventually she did actually eat honey. I was like, yeah, it's just bee. Okay. EPO, erythropoietin, is a hormone. Okay. You may have learned about EPO in anatomy. I also talked about it briefly before, but just let's reinforce it. EPO is released from the kidneys in response to hypoxia or low oxygen, and it makes you make more red blood cells. So where does it target? Yeah, it targets the red bone marrow. So hypoxia, right, targets the red bone marrow to cause erythropoiesis. Does that make sense? Okay, so it's just a little negative feedback loop like everything else has. And so that's called erythropoiesis. And your body produces on a regular day 2 to 3 million red blood cells per second. What? And so let's talk about the reasons that would cause you to release EPO, right? So why would you be hypoxic? Well, there's some good reasons and there are some not good reasons. So let's say it's January 2nd and you start exercising, but it's March and you're still exercising. You're giving a consistent error signal that says, I need more oxygen. And so what happens is after about 2 weeks of doing this, generally, you say, okay, I trust you that this is going to be your new pattern. And then your kidneys say, okay, I'm going to release some EPO and you're going to get some more red blood cells. So athletes actually have more red blood cells than the rest of us. Okay, so especially cardiovascular athletes. Other reasons that you might have release EPO. Increased altitude. So me, when I moved from Texas to here, after I'd stayed for 2 weeks, right? You get this consistent error message, and then eventually your kidneys go, okay, you're staying, Melanie, and then they release EPO and make me make more red blood cells, right? Or COPD, which is chronic obstructive pulmonary disease, which is just like your inability to, from some obstructive reason, you can't get enough oxygen in, so you end up being hypoxic. Or what about giving blood or hemorrhaging? Yes? Immediately after you gave blood or immediately after you hemorrhaged, right, your kidneys are going to go, hey, you need more blood, and they're going to release it and target your bone marrow, right? Also, destruction of red blood cells, like with malaria. Do you guys know about malaria? Malaria is an intracellular red blood cell pathogen that eventually causes hemolysis during its life cycle, basically to put out more little malaria pathogens to infect more red blood cells. And so when a person is infected with malaria, does it make sense that they're going to be hypoxic? Yeah? And they're also going to be freaking exhausted, okay? Really, really exhausted. Okay, so that brings us to our next one. EPO has now been genetically engineered, meaning that we figured out the DNA sequence and figured out actually how to make it in the lab, right? So now we can make it, and it's a good treatment for instance, people who are going through chemotherapy. More red blood cells would help, right? But it's also, of course, been abused because cardiovascular athletes would not like a competitive advantage over the other athletes. And so blood doping is a thing. There's a couple of different ways to do blood doping that I know of. One way is I can give my blood, keep it in the fridge, right? Give myself time to regenerate the blood volume that I've already donated, right? And then give it back. Just the red blood cells, right? Okay, that's blood doping, okay? The other way is to shoot yourself up with erythropoietin that you just bought, okay? Either way increases the likelihood of what's called erythrocytosis, which is too many red blood cells. And so you can have healthy cardiovascular athletes that, for instance, have increased their viscosity so much that they die of heart attack or stroke. That's called erythrocytosis. One of the ways that they've counteracted that is, I don't care how. So they couldn't figure out, like, okay, so it's your blood cells. How do I know how you got so many? Because you're an athlete anyway, right? Or by the time somebody tests you, the EPO is already gone from your system, right? It's gone because you did it weeks ago because you were going to compete today, right? So now what they do, the last time I checked last year, was in cardiovascular sports where blood doping is a problem, cycling. Lance Armstrong was busted for blood doping. What they do is they say, I don't care how you got higher than this outrageously high level. If your red blood cell count is higher than that, you can't compete, right? Regardless of how you got there. And it's a little lower for women than it is for men, and I'll tell you why in just a second. By the way, just a tidbit. Guess how we used to get EPO before we figured out how to make it in the lab? Crazy story. No. We bought the urine of malaria patients from other countries. And because they made so much of it that they just ended up peeing out the EPO, and we extracted it from their urine and used it for treatment here. I really, really hope that we also gave them malaria treatments, but I'm a little cynical in my old age because I'm afraid perhaps we did not. But that's where we used to get it. Go ahead.
If you're getting a blood transfusion from someone who has like crazy high EPO numbers or like platelets, would it make a difference?
No, I mean, initially it would, but eventually it wouldn't, right? Wouldn't really make much difference eventually. Okay, the other thing that can cause EPO secretion, and does, is testosterone. So testosterone forces the kidney to make EPO better than estrogen does. So typically speaking, males have not just more blood, but their blood is slightly different than females. In a cubic millimeter, which is about the size of a big drop, females have around 4.5 million red blood cells per drop, and males have almost 1 million more. So they have greater oxygen carrying capacity because of testosterone. that make sense? So again, as you get to the elite levels of cardiovascular sports, males and females don't compete against one another. They have greater muscle mass, bigger body size, greater oxygen carrying capacity. They will, however, ladies, die first. So comes out in the wash. Go ahead.
About this because they actually test trans females now for their testosterone levels before they're allowed to compete, and there's of course on that. There's an interesting article out that I could share, which is like a view of one trans female saying it's fair, and a view of one trans female saying it's
Oh, yeah, I'd like to read that article.
Discrimination. It's kind of
Well, if you're competing in athletics and you're competing against people with X amount of testosterone and therefore X amount of oxygen carrying capacity, then it seems like reasonable that you should be within the range.
yeah, I don't know. I don't have a view on it yet. It's just interesting. Like, are you recording?
Yeah, I've been recording. There's no room for it. Yeah, I want to see it. So anyway, that's testosterone. Okay, so now before, do you guys remember that red blood cells are anucleate? They don't have nuclei once they're mature. So before they actually eject their nucleus, they have to make the hemoglobin, right? Because you can't do protein synthesis without the code, right? Okay, so before they do that, they make hemoglobin, and hemoglobin is, of course, what makes your blood red and carries your oxygen. Note to self, when oxygen binds to the heme group of hemoglobin, it's kind of like rusting your blood, so the blood becomes bright red, right? When oxygen is dropped off, it becomes less red. Although my 12-year-old does not believe me that venous blood in systemic circulation is not blue, it's true. It's not blue. It's less red is what it is. And he just doesn't believe. He actually went and asked my husband. I told him that blood was not blue, and he went and asked my husband. My husband's like, what the hell do you think I would know this for? My husband, who has still never seen a cadaver and cannot get blood drawn without passing out. And was a philosophy major in college. So it's not like he knows about this crap. He knows some interesting stuff, though. Okay, so males and females, because of the different numbers of red blood cells, have different amounts of hemoglobin. That should make sense to you. Okay, so what do your red blood cells do? Just big picture. They are gas fairies. Okay, here's what they do. And we're going to talk about this again with respiratory system. As they go through the pulmonary capillaries, gas moves down concentration gradients-ish. So oxygen is just going to move from the air and then get picked up onto the hemoglobin, right? And then the blood gets bright red, right? Okay. And then carbon dioxide is going to move from the blood into the air, right? And then of course you're going to pump it back to the heart and then pump it to systemic circulation. And then think about my little fairy, right? I am carrying oxygen, but I'm not carrying carbon dioxide to any great degree at this point. So what happens as I get to systemic capillaries is the tissues are low in oxygen because why? Yes, thank you, Anna, you're my favorite. Okay, cellular respiration, they're low in oxygen. They're also high in carbon dioxide because why? Cellular respiration. Hell, people, it was like a softball. Cellular respiration. So what's going to happen is you are going to, oxygen is going to move down its concentration gradient off the hemoglobin. Carbon dioxide is going to move down its concentration gradient into the blood. And hemoglobin actually can carry oxygen and carbon dioxide. And it can carry them simultaneously because they don't sit in the same seat on the bus or the ferry. So here's where oxygen sits. Only 4 seats for oxygen, okay? But hemoglobin can travel on the globin portion, yes? So could there be a moment in which you were carrying oxygen and hadn't dropped it off yet, but the hemoglobin got on the bus? Yeah, there can. So it can actually carry both of them simultaneously. So when oxygen is bound to it, it's bright red, and it's called oxyhemoglobin. And when it's not bound to it, it's called deoxyhemoglobin. And you just sort of like do that over and over again. Okay, so you do carry a little bit of carbon dioxide on the globin portion, but only about 20%. Most of it is carried as the bicarbonate ion, which we'll cover again in the next set of notes, HCO3 minus. That's how most of it's carried. Okay, so red blood cells don't last forever. They get the crap beat out of them as they're going through circulation. And what happens is every time they pass through the spleen and the liver, you look to see if they've got one more round left in them. And if they don't, you just recycle the damn things. And then you keep all of the really important parts, the amino acids from the globin, you dump into bloodstream so you can use them for whatever you want, right? And then you dump the iron into the bloodstream so you can send it back to the bone marrow. Does that make sense? So you're really just recycling this whole process. Okay, So if we have time for clinical connections at the end, I will do anemia. If not, I'll just post a little 10-minute lecture over it because I want to get to platelets and hemostasis. Okay, so what we are going to do now is talk about hemostasis, which is not the same as homeostasis. So break it down. What does hemostasis mean? Hemo, blood. Stasis, stable, right? Stabilizing, okay? So what hemostasis is stopping the loss of blood when you're injured. It includes clotting, but it is not clotting, okay? And hemostasis and homeostasis look very similar, and I will put both of them on a multiple choice question, right? But you have to pick the right one. Is hemostasis homeostatic? Well, yeah, it totally is, but it's not the same word. Okay, so what hemostasis is, big picture understanding here, is all of the stages that allow you to stop blood loss and heal a damaged vessel, okay? And it has 5 stages. So I want to go over the big picture of the 5 stages first, and then we'll go into the detail. You ready? Okay, so I have damaged a vessel. I've either done an injection or had an injury, whatever. And then I'm going to do 5 stages. So the first stage, let's go through these all, and then we'll go through them in detail. The first stage is just vasoconstriction, and that should make sense to you. Vasoconstriction is just zhit, and do you want to vasoconstrict upstream of the damage or downstream? Upstream, yeah, right? I mean, you've had a hose before. If it's got a hole in it, you kink it upstream, right? So that's vasoconstriction. We'll talk about how it happens in just a second. The second phase is platelet plug formation, in which your platelets start to congregate. This is not a clot, okay? Platelets start to congregate in the site of damage, right? And if it's just a little bitty hole, they may plug it effectively, but it's not the same as a clot. So this is platelet plug formation. So they're just going to start to congregate in the area. And then, if that wasn't adequate to plug the hole, you will do the next phase. And the next phase is forming a clot. This is called coagulation. Clot is all of these insoluble proteins that were not present before. Think spider webs, right? So they're kind of sticky and they catch almost anything that goes by in the area. And what they do is they will form a little gel that plugs the hole. This is not the same as a platelet plug, okay? So this is a clot or coagulation. So I just did the first 3. Vasoconstriction, kink the hose, right? Did I do 3 or 2? Oh, platelet plug formation, congregate the platelets. Coagulation, form the spider webs, right? And then the next 2 phases are, and we're going to come back to them in just a second, clot retraction. One of the things that happens, and we'll look at the mechanism in just a second, is that when the clot forms, it's sort of attached to the wall of the damaged vessel. And then when it's about healed, it retracts to pull the edges of the damaged vessel close together so that it's easier to heal. That's clot retraction, okay? You have experienced clot retraction if you've ever had a road rash and felt that moment in which it like pulls together and it kind of squeezes out that clear fluid, right? That's clot retraction. And then the last phase, there's 5. The last phase is called fibrinolysis. It's dissolving the clot. The clot is made of fibrin, and so you do fibrinolysis. Okay, so one more time, the 5 stages, and then we'll go through each one. Vasoconstriction, that's how you start. Platelet plug formation. Coagulation, this is the complicated one. That's the one that everybody hates. It's not as bad as it originally seems. Coagulation, which is clot formation. Clot retraction, and then fibrinolysis. Okay? All right, so let's get started. So the 5 stages of hemostasis, the first stage is really easy. It's just vasoconstriction. Vasoconstriction happens in every vessel except a capillary. Anybody want to tell me why you can't vasoconstrict in a capillary? No smooth muscle, exactly. So any vessel except a capillary can do vasoconstriction. Got it? Okay. So the smooth muscle in the wall constricts. It lasts for around 30 minutes because you don't want it to do this and then open back up, because what do you have to do before you open back up? You have to clot and you have to fix the hole, don't you? You have to plug the damn hole, fix it, repair it. Okay. So the cause, I put a question mark there because we're still not completely sure about why vasoconstriction occurs. We know that partly it's because of paracrine agents are released on damage, and those paracrine agents that cause vasoconstriction include weird ones. Like ADP is released as a paracrine agent and causes vasoconstriction. Serotonin is released as a paracrine agent and causes vasoconstriction. And then another one called thromboxane A2. But they also think that possibly the nervous system is helping. It's like a nervous response to injury, but they've never been able to confirm that. So that's why I put a question mark. So we're not exactly sure why vasoconstriction occurs. Okay, the next phase is platelet plug formation. And it's basically a platelet party. So here's what happens with the platelets. Here's your platelet party. Okay, I always say that platelets are like teenagers. You have to be careful about letting them congregate unsupervised. Because when you let platelets congregate unsupervised, you're going to form a clot. And when you let teenagers congregate unsupervised, you're going to form a baby. At least in my high school. So what happens is whenever platelets start to gather, they start to send out a group text and attract their friends. It's a positive feedback loop, right? And then they send out a group text and attract more friends, right? And eventually you've got a whole bunch of platelets in one area. And if you let them sit there for a minute, they're going to start a clot even if there wasn't a hole, okay? So you got platelet party, platelet party. I love that one. It makes me so happy. Okay. All right. So here's what a platelet plug looks like. Do you see this? Just a whole bunch of little platelets. The way that it usually starts is this, okay? This is how it usually starts. If a platelet encounters a damaged vessel wall, and one of the ways that a platelet can tell that something is damaged is that a platelet encounters collagen. Platelets shouldn't usually encounter collagen because what is the lining of a blood vessel? What lines a blood vessel? What kind of tissue? It's just slippery epithelium. So if it comes into contact with a collagen fiber, it says something is amiss here. Yes? Okay. So if it comes into contact with a collagen fiber, then what it's going to do is it's going to say, whoa, and stop right there. And then it's going to stick to the collagen fiber and then send out a group text to its friends, right, and start attracting more platelets. The nature of the group text is primarily the release of paracrine agents that will attract any platelets that are going by to the area. And they include a couple of things. Again, ADP also does this. Thromboxane A2 does this. There's some more, including prostaglandins, epinephrine, serotonin. All of these are just positive feedback loops that attract other platelets. And then this one that I just want to tell you about, even though it's not in the image, because it's got some cultural significance. This one is called von Willebrand's factor. It's a paracrine factor that causes a little positive feedback platelet attracting loop. And von Willebrand factor binds to collagen, activates the platelets. When the platelets, by the way, get activated, what they do is they become sticky, but sticky really just means polar. What happens is they get a positive, all of their ions are positive at one end and negative at the other end. And so what happens when a platelet goes by? Just attracts it, right? And then it makes the other one polar. So you've got all of these activated platelets. They're kind of polar little cell fragments. The reason von Willebrand factor is kind of interesting is because if you can't make von Willebrand factor, you're missing one of the paracrine agents in this whole loop, which can really slow platelet plug formation, which can cause clotting problems, which causes one of the many, many forms of hemophilia. The reason this one is worth mentioning is because this one was rampant in European royal families and killed off a whole bunch of monarchs and a whole bunch of potential heirs to the throne because it was a genetic autosomal sex-linked recessive disorder. But when you marry your first cousin and have babies, it's going to end up being really common in your lineage. Does that make sense? So you guys know the European royal family had massive amounts of inbreeding and hemophilia. So a bunch of royal babies died off because of von Willebrand's hemophilia. It's kind of interesting. Okay, so these activated platelets, meaning sticky platelets, will start attracting their friends by releasing all kinds of paracrine agents. And eventually what happens is you get this platelet plug. And the platelet plug could be big enough to fill a little bitty hole, like just an injection site or something, right? So it can stop bleeding in a small injury in about a minute, okay? So question for you. If this is positive feedback, what could happen is you continue to attract and attract and attract, and it fills up the whole damn thing, right? So what localizes the platelet plug to just areas where damage has occurred? It's because the healthy tissue is constantly saying, get off my lawn. So this is teenagers, right? Empty house, right? This is the neighbor. Get off my lawn. So what happens is healthy endothelial cells release things that are anti-platelet plug formation, right? They say, do not stop here, okay? And what that does is it sort of sequesters the platelet plug to the area of damage. Good? Okay, so the platelet plug is not the same as a clot, but often precedes a clot, and we'll see why. More platelets, more likelihood of a clot. Let's briefly talk about aspirin therapy. What aspirin therapy does, they always call it a blood thinner or an anticoagulant, but it's actually not. What it is an antiplatelet plug former, because what it does is decreases the amount of prostaglandins in the area, and you lose one of your paracrine agents, so it makes it less likely that you'll form a platelet plug, and therefore less likely that you'll go the next step to form a clot. I'm supposed to be on daily aspirin therapy, as are a lot of people with cardiovascular disorders. And it makes it less likely that you'll form an inappropriate clot because I've got atrial fibrillation, right? So why the hell do I not take it? Well, first of all, I'm terrible at forming habits, non-compliant and just generally pissy. But in addition, also, every time I run into something, huge bruises because it doesn't clot very quickly, right? When I'm on daily aspirin therapy. I should be on it though. So that's what aspirin therapy does. Okay, now we are to the part where it gets a little tricky. It's going to seem outrageously complicated and then it will clarify and you will be totally fine. So don't worry about it. Okay, so what we need to do is we need to get from this phase to, where the hell is it? To this phase, right?
When the pellets will know when to stop coming.
What?
When, like, this phase will know that it's finished, that it, I can say that word, that's it.
Platelet. Mm-hmm.
It will finish.
That's going to happen as we finish the whole clotting process, or as the healing, as it no longer comes into contact with the collagen fiber, right? If it's no longer exposed to collagen as you heal the wall of the vessel, then you won't activate the platelets anymore, right? Okay, so what we need to do is we need to get to this stage, which is coagulation, and you form these insoluble protein fibers like spider webs that catches everything that goes by. Note to self, these these red blood cells are not part of the clot, they just got caught in the clot. I mean, they didn't make the clot, but they got caught in the clot. These are platelets right here, this one and this one. Those probably started the activation of this clot. Okay, but just everything that goes by, just like a spider web, gets caught in it. Okay, so here's the deal with blood. If you let it sit, it'll form a clot even if there wasn't a hole. That's why atrial fibrillation is a big deal, right? Because if you let platelets congregate, eventually what they do is they sit around and they go, hey, you want to form a clot? I want to form a clot. Let's form a clot, right? And then they just form a clot. There wasn't a hole. There was no need for another person on the planet either, but they made a baby, right? So they formed a clot. So what happens is, have you ever like seen a drop of blood on a table and seen the fact that it kind of like forms a little gel, right? And it actually pushes out serum at the edge. Have you ever seen that? If you let it sit there for just a second, it'll form a gel and then it'll actually push out serum at the edge. It's because all the clotting factors stay in, and serum is just blood plasma minus clotting factors. So basically, you don't really have to do anything to get blood to clot. You just have to not let it flow. So the gel is a clot made of the protein fibrin. So think about this kind of like Jell-O, right? Because what Jell-O is gelatin that's primarily formed from, this is so gross to think about, and it will make you never eat Jell-O ever again in the history of your life. It's formed from the collagen fibers of the connective tissue of animals, right? That you melt and then allow to gel again, right? And then these are little chunky things stuck in the Jell-O, right? My child was absolutely mortified when I told him what Jell-O was. He was like, ah! Okay, so, oops, what happened? Come back. Okay. What's that? Oh, turn that off if it's yours. So the clotting is what it is. The clotting is a cascade reaction. So stick with me for just a second. What you have is you have an inactive protein that gets activated, and then this one makes one that activates the next one, and this one makes one that activates the next one, until ultimately at the end of the cascade, you get the spider webs, which is fibrin, okay? So it's called a clotting cascade or a coagulation cascade. Does that make sense? Okay, so it involves a whole bunch of different factors. Most of them are made by the liver, okay? What is happening?
The South City campus is open. It was closed this morning.
Oh, is that what that is? Okay. So it's a domino effect. Most of these are made by the liver, and most of them are present all the time, but just not activated. So look here and don't freak out when I show you this, because I'm going to simplify it for you, okay? Okay. So what you have is a whole bunch of clotting factors. One of them gets activated and then activates the next one, and that one gets activated and ultimately makes fibrin. Before we look at the figure, let me tell you something. Each clotting factor is denoted by a Roman numeral, like factor X is actually factor 10. When it gets activated, it's 10A, active 10. Got it? 12, 12A, okay? So look at this figure for just a second. This is where we're trying to get, right? Fibrin, okay? Do you see that what happens is you activate this one and it activates that one? You activate this one and it activates that one, right? Till you get all the way down to the bottom. There are two different pathways you can take to get there, okay? I do not have you memorize every single part of this. I have you memorize the important parts, which is the end, duh, right? The middle, because once you get here, you're going to get a clot no matter what, and the difference between these two pathways. Does that make sense? Okay, so there are a whole bunch of different factors involved in this. Most of them are made by the liver, and most of them are sitting around inactive all the time. Any idea why you would want these factors to sit around inactive instead of just making them upon damage? Yeah, exactly. Because do you want to fart around with blood clotting? No, you want to get it done really quickly, right? So they're just sitting around inactive, and then you can activate them and it'll activate the next one. So note to self, the weird thing about this is that all of these are not numbered in order that they act. They are numbered in the order that they were found. So it doesn't help anybody at all. So you got like 10 down here, and then you got like 2. Right? You're like, thank you. So for that reason, I don't usually have you memorize the numbers except for this one that's the important branch point right there in the middle. Okay, so clotting is this entire process and usually takes between 3 and 6 minutes. So about a minute for platelet plug formation and then about 3 to 6 minutes for clotting. And so it's going to require calcium at multiple points. So remember, I've been telling you all semester long that calcium is required for a whole bunch of crap. Now we've seen examples it's required for neurotransmission, right? Exocytosis, required for cardiac muscle contraction. I told you it's required for clotting, but look where it's required for clotting. Look how many different places. 1, 2, 3, 4. Calcium is intimately involved in the clotting process. And if you don't have calcium, you're not going to clot. Okay, and this is extracellular calcium, of course, so it's regulated primarily by PTH and calcitonin. So, all right, so keep the calcium thing in mind for just a second. Okay, so here's what we're going to do first. Do you see that once you get here, you get a clot? Okay, so we're going to do this part first. This is called the common pathway. Once you get here, which is factor 10A, I'll explain it to you in just a second, you're going to get a clot regardless. There are 2 possible ways of getting there. So what I'm going to do is I'm going to come back and pick up those 2 possible ways. All right, so 3 stages in the formation of a clot. The first stage is getting to here, and the second stage is converting that one to that one, and then that one to that one. So let's go here, which is factor 10A. So 10A is one of the few that you will have to know by number and by name because it is a really important part of the story. Because once you get 10A, you're going to get a clot, as long as you have calcium, okay? So 10A is also called prothrombinase, and it doesn't say that on this figure. By the way, I have a figure that really simplifies this stuff for you, and I forgot to post it before class, so I'll post it after class for you. It's just like the same thing in a lecture, actually in a little figure, okay? All right, so prothrombinase. Here's what prothrombinase does. Prothrombinase, so what does pro mean? Before. Thrombo always implies what? clot, yes? And ASE is an enzyme. Here's what prothrombinase does. It converts prothrombin before thrombin, yes, to thrombin. Before I go any further, look how logical that naming convention is. Before thrombin to thrombin is by prothrombinase. Okay? But we're not at fibrin yet, and that's where we're trying to get, right? Okay? Thrombin converts fibrinogen, genesis as in beginning, to fibrin, which is stabilized to form a clot. Okay? This is just fibrin stabilizing factor, which forms clot. Okay. Before we go any further, which of these factors are always around? Prothrombin? No, prothrombinase is not always around. Prothrombin is always around? Fibrinogen is always around. But do they ever interact with one another? Unless you start the whole clot formation, right? Okay, so we've seen examples like this before. There are a whole bunch of things around all ready to start a cascade, but you need a trigger, right? With the renin-angiotensin-aldosterone system, we saw that, okay? So these two don't interact with one another. So let's find this on the figure real quick, and then we'll go back and figure out how you got there, okay? All right, so you with me? Okay, 10A, which is what? Prothrombinase, converts prothrombin to thrombin, good. And then thrombin converts fibrinogen to fibrin, which gets stabilized to form a clot, and that's your spider web. Got it? Okay, so this is called the common pathway of coagulation because it doesn't matter how you got there, right? Okay, then we'll go back and figure out how you started. How did you get to prothrombinase? There's two different ways of doing it. Prothrombinase can be formed in two different ways. You can do, so let me just tell you the names and then you'll figure out which one is which. You can start it with factors that are circulating in the bloodstream, okay? Or you can start it with a damaged tissue or vessel wall, okay? Now you put the names to each one. Which one are you going to call the intrinsic pathway? The one that starts from within the bloodstream or the one that starts with damaged tissue? Within the bloodstream, yes? And then the extrinsic pathway starts with damage to the tissue or the vessel wall. Extrinsic pathway, right? Good? Okay. So you've got intrinsic pathway and extrinsic pathway. You guys understand that? Okay. So the extrinsic pathway, let's do that one first. Okay. The important thing that you need to know about the extrinsic pathway is that a damaged tissue will ultimately cause the release of something called tissue factor or tissue factor 3. Do you see it? Tissue factor? Okay, and then the tissue factor will ultimately cause the production of prothrombinase, factor 10A, okay? So just keep that in your head for just a second because now I can tell you that the intrinsic pathway starts with a platelet party, okay? If you let the platelets congregate, eventually they will start to release something that eventually gets you platelet factor 3, PF3, okay? And platelet factor causes the conversion of 10 to 10A, and then you get prothrombinase. So, clarify. Extrinsic pathway, tissue factor, because of tissue damage. Yes? Okay. Intrinsic pathway, platelet factor. Starts with platelets. Got it? Okay? Okay, so before we go any further, let me ask you a concept check question. Which one of these pathways is probably going to get you inappropriate clot formation with no damage, like with atrial fibrillation or with arteriosclerosis? Yeah, do you see? It's the intrinsic pathway. And what's the problem? If you let them hang out long enough, they're going to start this process regardless of whether there was a hole. Yes? Okay, so it's the intrinsic pathway. Now, if you actually damaged a vessel, right? Decent amount of damage to the vessel. Which one do you think is going to be activated? Both of them, yes. Both of them are, right? So if you activate both of them simultaneously, then you just get faster to the common pathway. Does that make sense? okay? But in a situation in which there wasn't any damage, then it's just this one, right? Okay. In the situation in which there was damage, it's probably both, right? Contact with collagen and the tissue is damaged. Does that make sense to you guys? Okay, so that's an extrinsic and intrinsic pathway. So the intrinsic pathway is initiated by chemicals within the blood. Those sticky platelets eventually sit there and go, you want to make a clot, I want to make a clot, and release ultimately platelet factor 3 and then start that whole process. Okay, and the common pathway begins, doesn't matter how you got here, you're still going to have a clot. Did you have a question, Anna? Oh, okay. Okay, so the common pathway. Okay, so now let's clarify and simplify. You ready? Okay, so all you have to do is read this, and it gets you most of the crux of what you're trying to get. So let me show you how to read it. The extrinsic pathway is activated by tissue damage, which releases tissue factor 3, yes? Which activates prothrombinase, that's where the common pathway starts. You with me? Yes? Which converts prothrombin to thrombin, yes? Thrombin converts fibrinogen to fibrin. Fibrin is stabilized to form a clot. Got it? And then let's do the intrinsic pathway. Ready? The intrinsic pathway is usually activated by an injured vessel with exposed collagen that causes sticky platelets. And then your sticky platelets release platelet factor 3. Yes? And platelet factor 3 activates prothrombinase. That's where the common pathway starts. And then it does its thing, which converts prothrombin to thrombin. Thrombin converts fibrinogen to fibrin. Fibrin is stabilized to form a clot. Right? So it clarifies, right? It's not as bad as it originally seems. Okay? Okay, so let's talk about a couple more things that are needed in this process. We've already done all the hard part. Okay, so there are at least 13 different factors that are needed for clotting. This isn't even all of them. Some of them are ions, some of them are proteins, most of them are made by the liver, okay? Most of the proteins are made by the liver. You're only responsible for any factor that I described by name or number in your notes. If it's not in your notes, I'm not going to ask you 9A right here, okay? So do you see the point that I tried to get you guys to was how do you start it? Where's the common pathway, right? What's the unique part of this pathway, which is platelet factor? Unique part of this pathway, which is tissue factor. And then where do you end up, right? Okay, so it requires calcium at multiple points. So which pathway uses calcium, guys? All three of them, right? Extrinsic, intrinsic, and common, right? They all use calcium. So if you do not have extracellular calcium, you are going to have clotting issues, right? Okay, so, and then the last thing I want to tell you guys about this before we go into the clot retraction phase is just the whole role of vitamin K in the whole clotting thing, just to sort of reinforce something that we've looked at before. So we learned vitamin K way back at the beginning of the semester. It's one of your lipid-soluble vitamins, ADEC, right? Okay. And vitamin K is mostly made by your intestinal flora and your large intestine, okay? And your intestinal flora, your natural intestinal flora will make vitamin K and then you will absorb it. So it's made by bacteria in the intestine. The important part about this is that vitamin K is needed for the liver to make prothrombin, okay? If you don't have vitamin K, you can't make prothrombin. And prothrombin is, of course, part of which pathway? Yeah, it's part of the common pathway, right? So you're screwed if you can't make prothrombin, right? Okay, so it's lipid soluble, made by the liver. You need bile from the liver to absorb it too. So let's go back to the whole vitamin K injections in newborns just to sort of reinforce what's going on here. So with newborns, which he probably has far more experience than I do. Ooh, I wonder, I'm gonna ask you about this. So with newborns and vitamin K injections, some brilliant person, it just occurred to me that I should look up who did that because that's pretty damn brilliant. Some brilliant person figured out, okay, so newborns, of course, haven't colonized their intestines yet because they haven't used their real digestive tract to much degree. So they've got a period of time right after they're born in which if anything happens to them, they're far more likely to bleed out than someone who is like 2 weeks old or 4 weeks old, right? So somebody figured out, well, we could protect them for their first about 8 days until they get colonized by giving them a vitamin K injection so that they could make prothrombin, right, in their liver. And because sometimes, especially if anything's happened to the baby, you're going to need to poke them, right? You might need to poke them and something might happen to them. Also, birth can be potential for damage during birth. So they do vitamin K injections, and it protects you for about the first 8 days of your birth. But then I think I talked about this before. What's interesting is that with the whole anti-vaccine movement, people are confused and think that anything that comes in a syringe is a vaccine, and people have chosen not to give their babies vitamin K injections. So you have unnecessary hemorrhaging in babies. Have you guys run into that in your work?
For babies hemorrhage?
Where people forego vitamin K injections?
Oh, yeah.
Oh, yeah, really?
Oh, yeah.
Wow, really? So the last time I talked to my OBGYN, who's a high-risk pregnancy OBGYN, he's also a research doctor, he told me that, and it was 5 years ago when I talked to him, so the anti-vaccine movement has really like bloomed since then. He told me, I think that what he said is during the last 5 years of his practice, he has had about 20 times more people foregoing vitamin K injections, and he hadn't really had any babies hemorrhage and lose them until about the last 5 years. So it's kind of tragic because, of course, it's just a gamble that nothing's going to happen to them during the first times. Have you lost any babies due to hemorrhage related to that? You haven't? Yeah. So anyway, it's just a vitamin. That's all it is. It's just a vitamin that they actually haven't made yet, and you can protect them. Okay, so that's coagulation, right? Okay, so tell me the 3 phases that we've gone through in order. Vasoconstriction, platelet plug formation, coagulation, and now the last 2. And the last 2, this one's so weird and cool to me. Apparently, these little buggers right here, in addition to being chock full of like platelet factors, clotting paracrine agents, they have actin and myosin in them. And when the tissue is just about healed, a paracrine agent is released that actually activates the actin and myosin. And the spider web is kind of like stuck to them and also stuck to the edges of the damaged vessel. And then it does just a little bit of a spasm. They can't do a whole lot of cross-bridge cycling because, of course, they don't have any mitochondria or anything, right? So they do just this little spasm that pulls the edges of the damaged vessel together that makes healing easier. And they just figured that out like 15 years ago or something, that like there's actin and myosin. They still don't quite understand exactly how the hell it works in there. But anyway, that's called clot retraction. And clot retraction is when you feel like if you have a scab and you feel it tighten up, right? That's the clot retraction. And that is, again, when quite often you squeeze out that clear yellow fluid. Have you guys seen that with road rash? When you get road rash and you squeeze out that clear yellow fluid, that's called serum. It is blood plasma minus all the clotting factors that stayed in there. That's what it is. If it's opaque, it's a problem because that means there's actually an infection. But if it's clear, it's generally just serum. Okay. And then the last phase is the fibrinolysis phase, okay, which should make sense to you. This is lysis of fibrin. So when I am done and I am healed and the hole is plugged and all is good, what is going to happen is you need to dissolve the clot to restore blood flow, right? Right? Okay, so what protein was the clot made of? Fibrin, exactly. So here's what happens is you start a different cascade when the damaged tissues start to heal. So healing tissues will release TPA, which is tissue plasminogen activator. It's a paracrine agent, okay? And TPA converts plasminogen, as in genesis or beginning, to plasmin, and plasmin dissolves fibrin. Does that make sense? So the tissue says, I'm good now, releases TPA, plasminogen, always around. I don't remember where it's made. Let's guess liver, probably. I don't know. So now you know that in physiology, it's cellular respiration, homeostasis, liver, hypothalamus. That's all the answers for the whole semester. So this one is always around, right? When you are ready to be done, you release TPA, it converts plasminogen to plasmin, and then plasmin dissolves fibrin, and then you're good. Does that make sense? Okay. TPA, clinically really important because it is a fantastic clot buster. Can you imagine that it would be? Yeah? It's also super duper expensive and not given as a maintenance medication. So you would have to be like inpatient to get tissue plasma, to get TPA. Okay. And why? Because if anything happens to you, you're going to bleed out, right? If you've got TPA in your system, because you're not going to clot at all. So it's been genetically engineered now. We know how to make it in the lab, and it's available in quantities large enough to be used therapeutically. They use it as a clot buster in strokes. Yeah? They use it as a clot buster in strokes. But it's so expensive that you're not going to get it if you don't have good insurance. It's like cost prohibitively, like thousands for a treatment, expensive. So I know it's like uncomfortable to think about, but people who do not have a good insurance get different health care than people who do have good insurance. And that's a little disturbing to think about, but it's true. Okay, so that's your clot buster. Everybody good? Okay. So clotting considerations is the last thing that we're going to do. Do you guys have questions? Didn't really stop yet for questions.
So do you have things like warfarin?
Oh, I'm about to talk about this. Okay, Coumadin and warfarin are right here. Okay, so we know what prevented a platelet plug from spreading beyond the site of injury, because the healthy cells were saying, "Get off my lawn," right? So what prevents a clot from spreading beyond the site of injury, right? Why, when you form a clot, does it not keep going? Well, it's kind of the same thing, with the exception of the fact that in your bloodstream, you always have anticoagulants that are always active, right? And they're saying, no, no, no. Where do you build up activated coagulants? Well, only at the site of injury, right? And then you're carrying them away, so you only continue to increase the concentration of coagulants at the site of injury. So the blood circulation carries things away except for the site at which they're constantly generating it. And there's a whole bunch of anticoagulants in the blood that are always around. Antithrombin, heparin. Heparin is secreted by mast cells. It's also secreted by basophils, and it's constantly in the bloodstream saying, don't clot, don't clot, don't clot. So really, clotting only occurs if you have a location in which the concentration of these activated clotting factors exceeds the concentration of the always active anticoagulants, which on a good day should only be at the site of damage. Yes? Okay. In addition, there's a little negative feedback loop. I don't usually ask you about this, but as fibrin starts to get really, really big, it actually inactivates thrombin. So as fibrin starts to be like, I'm a whole big clot, it's like, you can chill, right? So it inactivates thrombin. Okay, so let's talk about heparin, Coumadin, warfarin. Okay, heparin is, you guys have probably run into heparin, right? In clinical situations, heparin is administered therapeutically to stop clotting during surgery because you don't want it to occur. They used to use it for IV locks all the time. Now a saline lock is more common, but they still do have hep locks. I think they use them for like PICC lines and stuff. Do you guys know this better than I do? You know I deal with dead people far more often than I deal with living people. So in a lot of what you want is, if you have an IV, for instance, you do not want clotting to occur around the IV, right? So they can use saline in some places, and in some places it actually has to release small amounts of anticoagulants, and that's heparin. Heparin is also what I had to use during my pregnancy because I had atrial fibrillation, right? And then estrogen increases the likelihood of clotting. So with those double risk factors, I had to shoot myself up twice a day during my entire pregnancy with heparin to make sure I didn't clot inappropriately. And then they had to keep me in the hospital afterward, after I'd had the surgery, because I had to deliver via C-section to get all the heparin out of my system so I would clot normally again. Okay, so that's heparin. And what heparin does is heparin destroys thrombin, breaks down thrombin. That's how heparin works, okay? Coumadin and warfarin. Okay, so Coumadin and warfarin are interesting. So I know you know somebody who's on Coumadin or warfarin. They're typically referred to as blood thinners, but they're really not blood thinners because they don't really change dramatically the viscosity of your blood. What they really are is anticoagulants, right? to decrease the likelihood of inappropriate clotting in somebody with hypertension or arteriosclerosis or atherosclerosis or some other predisposition to strokes or heart attacks or whatever, right? Or maybe somebody that already had a stroke or a heart attack and you're treating them with this to prevent another one. Okay, so what these do is they are vitamin K antagonists. How the heck does that work? If you take out the vitamin K, then you can't make what? Prothrombin and therefore thrombin, right? And you can't start the common pathway. You can't activate the common pathway. But of course, what happens to these people if they run into the coffee table? They bruise like crazy, right? Does that make sense?
Would they bleed too?
A lot of times, yeah, they can bleed. If you get damaged, they're going to have a hard time. Like nosebleeds are really common when you're on Coumadin and warfarin. If you damage something, you're going to bleed longer than other people. So it's a vitamin K antagonist. Hopefully it doesn't take out all of the vitamin K because then you won't clot at all. So they have to really regulate the amount of Coumadin and warfarin. Does that make sense? Okay. In addition, you also have to change what you eat if you are on these, because if you're taking a vitamin K antagonist but you're eating foods that are really rich in vitamin K, you're undoing the work. So you can't eat a lot of leafy green vegetables, salads, spinach, those kinds of things. It's pretty hard for somebody with cardiovascular disease anyway, right? Which they should be eating more of that. Okay. And then here's an interesting one. Why does your blood, if I give blood, why does it not just clot up in the bag? Some brilliant person figured out that in order to prevent it from clotting, all I need to do is add a detergent that ties up the calcium. Yeah, and it works, right? Okay. It's EDTA is one of the most common ones. All it is a little detergent that actually attracts all those positive ions, and then you can't clot. Okay. And it's totally fine because then if I gave my blood to you, assuming we were same blood type, what would happen is you just squirt it into him and he's got enough calcium and you can just like reactivate the normal clotting process. It's pretty cool. That's how you prevent it, right? You have to actually add something to keep it from clotting in the bag. Okay. So briefly, as you guys are leaving, so I will put the rest of this on a little video for you guys, but sometimes, of course, you do form a clot when there wasn't a hole. Yes? Okay. And that's sort of thrombosis. And if the clot is actually made of fibrin, right, then you can use these mechanisms to like get rid of it, right? But the blockage, the problem is not forming the blockage. The problem is circulating the blockage. So most of us, by the way, at some point or other are going to form clots in our legs, right? Because we've got low pressure running against gravity, right? And so a lot of times they form around the bowels and the legs. But that's all well and good because usually your anticoagulants will break them down. They'll just be relatively small. When they become problematic is when they start circulating, right? And they're going to circulate, of course, back through your inferior vena cava back to your heart, right? And it's okay if they're going through the chambers of the heart because the chambers are nice and wide. But it becomes problematic if, in combination with clot formation, you also have this narrowing of the artery because of atherosclerosis. So I want you to imagine that that's my anterior interventricular artery, or the LAD, or the widowmaker, right? That guy right there. So in a healthy person, if you form a clot and start circulating, that clot's going to go straight through the vessel and get broken down by the anticoagulants. But in a person with arteriosclerosis or atherosclerosis, what's going to happen is it's going to go down that vessel and it's going to get stuck, right? And when it gets stuck, it's going to cut off oxygen to your cardiac muscle. The same thing can happen in your arteries going to your brain, right? So that's one of the major forms of heart attack and stroke. And it's really a combination of clotting and arteriosclerosis or atherosclerosis, right? That's the lethal combination. Of course, both of those increase in likelihood with heart disease and with high fat, high sodium diet in combination with low levels of exercise, right? So it's like the Venn diagram of cardiovascular disease leads both to arteriosclerosis and increases the likelihood of clotting, right? Okay, so we will stop there. I'll post like a short 10 or 15 minute video to finish this up, and I'll also post a little diagram for you to practice this stuff on, okay? Have an awesome Thanksgiving. Oh, by the way, leeches work because they release heparin. Yeah, their saliva has heparin in it.